Continuing with the biotechnology series, I, Dr. Charu Dogra Rawat, Associate Professor from Ramjas College, University of Delhi, will talk about advanced tools and techniques, in which particularly I talk about the blotting. The learning objectives for today's session would be to understand the principle behind the blotting techniques and to understand the procedure of southern and northern blotting. If the time persists, we will understand the procedure of western blotting as well, else we will take it up in the next session. The references for the session particularly used for educational purposes is from T.A. Brown, the Gene Cloning and DNA Analysis, an Introduction, the 6th edition, and Twyman, Advanced Molecular Biology, a concise reference. We have been talking about recombinant DNA technology in this series. Recombinant DNA technology is the technology used to create a recombinant DNA. Now, when we talk of recombinant DNA, we combine DNA from two different sources. Now, as you can see in the other sessions, one of the DNA is the source DNA for a gene of interest, which is also called as the insert, which we put it into the vector. Another DNA which is called as the vector, because then it takes this insert, takes it inside a bacterial or a suitable host and makes it replicate, for example, for the purpose of molecular cloning. So, recombinant DNA technology, the basic step, the first step was to create this recombinant DNA molecule. As I said, there is a fragment of DNA. Now, this fragment of DNA can be commonly called as the gene of interest, although it can be a region also, including one gene or more genes. But this is the gene of interest, termed as the insert, which is then cloned into the vector. So, we have looked at the entire procedure of the different types of cloning vectors, the source DNA, how they both combine together. So, we have talked about the restriction enzymes, we have talked about the modifying enzymes, and then how they can be ligated to create this recombinant DNA molecule. We have also talked about how this recombinant DNA molecule is then introduced into a host, a procedure which is called as transformation. If it is done, the naked DNA is introduced into a bacterial host. It is called as transduction. If viruses are used to introduce this recombinant DNA into the host molecules, or it is also called as transfection or conjugation, in which the two host molecules, they combine together. So, one carrying the recombinant DNA molecule and the other to which it needs to be transferred. So, we have talked about all these methods. Then at the end, we also talked about how to assay the cells that contain the recombinant DNA molecule. So, we talked about the colony hybridization, we talked about plaque hybridization assays and all those kind of things to cover the entire recombinant DNA technology. Now, as this session is called Advanced Tools and Techniques, some of the techniques we require so as to do further manipulations, the genetic manipulations required for the biotechnological purposes. So, one of the requirement which is required for the recombinant DNA technology is the identification of the insert. Now, when in this figure, I am showing that there is a vector. So, vector is basically a plasmid that I have isolated plasmid or cosmid or bags and yaks and packs, all different type of cloning vectors that we had covered. So, we isolate this, and where do we get this gene of interest from? Now, this gene of interest will be present in another organism, for example. So, in one of the genome, we just want to know more about or make clones of a particular fragment. Now, amongst that entire genome, how do we find this fragment? So, this identification of the insert that we need to put in the vector to create a recombinant DNA molecule, for this purpose as well, the two techniques can be broadly used. So, one of the technique is called as the blotting and hybridization, which we will be covering today. In this, the genomic DNA, very broadly, the procedure for it is that the genomic DNA, which contains our gene of interest, is partially digested. We do agarose gel electrophoresis. We have already covered this visualization of DNA in that session. and then we blot on to the nitrocellulose membrane, basically nitrocellulose or nylon membranes, we will look at that, but we blot on this gel containing the partially digested DNA onto the membrane, and then we probe for the gene or region of interest. Now, probe is the word which basically means to investigate. So, amongst all the fragments that are created by the partial digestion of the genomic DNA, we try to investigate where is or what is our gene or region of interest that is located, because we are interested in only that region or that gene. So, we need to then identify it, isolate it, and then go ahead with the recombinant DNA technology. Another method which we will cover in our subsequent session, the technique is termed as the polymerase chain reaction or PCR. So, in PCR, the primers are designed for the gene or region of interest, and then the genomic DNA, from the genomic DNA, our gene or region of interest is then amplified. So, we will talk about this more in detail in our subsequent sessions. Today, we will be talking about the blotting technique. Now, what is the principle behind this probing and amplification? It's a common principle, and the principle why or how we can do it. So, how can we investigate a gene of interest or how we can amplify the gene of interest? The basic principle or the characteristic of DNA structure that is utilized in it is termed as the complementary base pairing. So, you must have heard we had covered this in our molecular biology series, the Watson and Crick double helix model of DNA. So, we all know that the DNA is a double-stranded structure. There is a phosphate backbone which is there are bases, nitrogenous bases, there are hydrogen bonds by which these two strands are joined together. Then these two strands are also anti-parallel. So, there is a 5' 3' orientation and there is a 3' 5' orientation. So, we have already talked about and discussed this thing. Again, this figure shows very clearly. So, these are the bases which are stacked one over the other. This is the sugar, and then there is a phosphate backbone. So, there is a phosphodiester bond between the two bases. This is one of the strand. As you can see, there is a polarity. It runs from 5' to 3' direction, and then the second strand is again anti-parallel. Now, these two bases are stacked together and joined together basically by this hydrogen bonds which are shown in the center. So, this is a typical Watson and Crick double helix model of DNA. The property which is utilized for the hybridization or blotting is the complementarity. Because there are 4 base pairs, we know that there is a restriction or there is a limitation that a particular base pair can hydrogen bond only with the particular base pair on the opposite strand. So, we had talked about this, that how did they deduce it? What was the reason that this is happening? So, during the discovery, there was Chargaff rule which was stating this thing. Then there was also some electrostatic possibilities that only this can happen. The C can only combine with G, A can only combine with T, and if there is a difference in these things, then it is not an electrostatically stable molecule that exists. So, now this particular thing that only C can make hydrogen bonds with G or A can make hydrogen bonds with T gives us what is called as the complementarity base pairing. So, if we know the sequence of one strand of DNA, we can always deduce the sequence of the second strand of the DNA. What do you understand by the sequence? Sequence is basically the arrangement of these nitrogenous bases. So, in what order do they come? So, they come as C, A, G, T, whatever. So, whatever this sequence is, then the other sequence we can always deduce it. So, one thing is that we can always deduce the other sequence. The second thing is, if we synthesize something, some oligonucleotide which is complementary to this DNA. So, this DNA is termed as the template DNA. This is the oligonucleotide which we synthesize or we take from any other individual, but if it is complementary and we incubate them together, then this complementary oligonucleotide will go and bind specifically to the region of the template which is complementary to each other. So, I hope, because this principle is very, very important to understand both the techniques that we have talked about. So, I repeat once again that if you know the order of sequence of one strand of DNA, and the other strand of DNA you can deduce. And if we also have another strand of DNA which contains the complementary base pairs. So what are the complementary base pairs? That the C will always combine with G, A will always combine with T. So when we have such a kind of an arrangement, then if we introduce an artificially synthesized oligonucleotide or a DNA fragment isolated from some other source but containing the order of sequence which is there, which is complementary to our template DNA, and incubate them together, then they too will tend to come together and create this hydrogen bonds. That is, they will create a double-stranded DNA. So this is the basic principle, the basic property of DNA that is utilized both for probing as well as amplification. Another important aspect or property of DNA that is utilized in these two techniques is what is termed as the denaturation and renaturation, also otherwise called as the melting of DNA. So when we change the conditions, for example, if we give certain high temperature, or we give rise to different salt conditions, or we change the pH of the medium, then these two double-stranded DNA which are joined together by the hydrogen bonds, they tend to fall apart and form single-stranded DNA. This is termed as denaturation. And when their conditions, the optimum conditions are restored, then they again tend to form the double-stranded DNA. So the DNA can be existing in the double-stranded form or a single-stranded form. There can be a denaturation and renaturation depending upon the alteration of the conditions. So these alteration of the conditions include the change in the temperature, the change in the salt concentration, or change in the pH. So this is what an important again property, the denaturation and the renaturation property. The third thing which is important, particularly for the case of amplification, is the formation of a heteroduplex, or in fact for both the things, which is the formation of the heteroduplex. So heteroduplex basically states that if there is a double-stranded DNA and if there is another complementary DNA which is present, which is not from this double strand but it has a complementarity. Now this can be a DNA strand or this can also be a RNA strand. Now I would like to remind you of the process of DNA replication. What happens in DNA replication? When the DNA needs to replicate, the two strands separate and the RNA primers, they come and bind to the origin of replication. If you remember, DNA replication cannot occur de novo. So there has to be a primer that comes, particularly by the DNA polymerase, one type of a DNA polymerase. It cannot exist de novo. So that RNA primer also binds. So RNA can also bind to the DNA template. The only difference is that the RNA contains uracil instead of thymine. However, the uracil can make the bonds with the adenine which is present in the template DNA. So the formation of heteroduplex is again very important, that it can bind to its own complementary DNA fragment, but if we give this complementary DNA fragment from outside, either we create artificially or if it is coming from some other organism, then it can still form this DNA and some other DNA strand or DNA and RNA hybrid. So this is called as a hybrid molecule. So three properties of DNA are very, very important. The complementarity base pairing. Second, that the DNA can denature as well as renature. And third, it can form heteroduplexes. Heteroduplexes with another strand of DNA coming from some other source, but it should be complementary, or even with the RNA. So there can be a DNA-RNA heteroduplex as well. So these three properties are very, very important principally to understand the procedure, both the techniques that we have mentioned in their earlier slides. So what happens in case of hybridization? So hybridization is basically that the two strands of DNA, they are separated, and then additionally either a probe or a primer, as I said, these probe or primer, the probe is generally can be synthesized artificially or it can be obtained from another organism also. So this can be a DNA fragment obtained from somewhere else. The primer, the amplified product, can also act as a probe. The primers are generally oligonucleotides which are artificially synthesized. So we contain this probe and primer. So when we mix them together and give certain conditions, then this primer or a probe can basically bind to its complementary region. So you can see this is the double-stranded DNA. So for example, this is the genomic DNA. We separate the two fragments of the DNA and we add the primers or the probe. So when we add the primers and the probe or the probe in two different techniques and we provide certain conditions, then this primer and probe can go and bind to the complementary region or gene in the genomic DNA. So this is how, it can easily now tell you from a total genomic DNA, you can identify a gene of interest with the use of these tools, which is basically the probe or the primer. So primers are also short oligonucleotides complementary to a part of the gene of interest. However, they are non-labeled. So we will talk about this more in the basic polymerase chain reaction. In the basic one, it does not contain a dye. It does not contain any marker to detect it, but it is basically a non-labeled thing. So labeled basically means that we add something onto the oligonucleotide so that it is something kind of a tagging, so that we can detect it later. So we will look at that procedure and you will understand it better. But the major difference between probe and primer, so probe is basically used in hybridization, primer is used in amplification. Probes are labeled, they are tagged with certain chemicals or certain radioactive molecules or something like that by which we can detect where the probe has bound. The primer, however, in the simple PCR reaction, they are non-labeled. So all they do is they keep synthesizing these molecules in various cycles. So we do not identify it by detecting it directly bound to the template, but because they create multiple copies of the gene of interest where they are bound. So this is the basic difference between a probe and primer, but I am generally talking about the principle of both the techniques which we do. So this is related to the hybridization. Because we will be talking about blotting in hybridization today, and I hope that you have understood the meaning of probe. And when I said that the probes are labeled, that means they are basically containing certain tags by which they can be detected after the technique has been done. So we will look at, before going into the technique of or the procedure, how these blotting techniques are done, we will talk about the probe labeling. So probe labeling is basically, one of the method is to do it with a radioactive marker. So with a radioactive marker we can use it. For example, here it is shown. So this is our DNA. This is our template DNA, and if we utilize DNA polymerase as well as we introduce the nucleotide which is radioactively labeled. So what will happen is this DNA polymerase 1 is going to fill these nicks or gaps which are there, but while filling we add normal nucleotides. So we add DGTP, DCTP, DTTP, DATP is what is which is radioactively labeled. So you can see P32 is attached to it. So wherever it is going to add this DATP, it is going to be radioactively labeled. So now we have tagged, we have primed this particular DNA because now it is giving a radioactive signal. Wherever the DATP is bound, now it is giving a radioactive signal. So when we use it as a probe, we single strand it and we utilize it to bind to a complementary DNA. Now wherever it is going to go and bind, then it is basically going to give rise to a radioactive signal which we can detect. So this is one of the method of probe labeling. Another method of probe labeling is, which is by the method of end filling. So this is an end filling mechanism. This is a nick translation method in which the nicks are created and then these nicks are filled with the synthesis. Another is called as the end filling when we have ends which are single stranded. So if you remember, we covered some of these enzymes and some of these things in the modifying enzymes. You can look at that session. When we were talking about the DNA modifying enzymes, we talked about this clinofragment. So this is the application of these modifying enzymes which are used to label the probes. So if the ends are single stranded, then the clinofragment has the capacity to synthesize or fill this end. So again we add the basic nucleotides as well as one of the nucleotides which is radioactively labeled. So these ends, for example, have been created by the EcoR1, a restriction enzyme which is producing the sticky ends, and these sticky ends have now been labeled to give rise to a radioactive tagging which is there. The third method is what is termed as the labeling by random priming. So in here we are randomly priming. So what we do, we make it a single strand and we add hexamers. So we just add random primers which is going to bind to these. So random primers because there will be random sequences, it is going to bind there, and again we add clinofragment and radioactive probe which is going to label this particular DNA. So this is again one method of labeling of the DNA by the random priming method. The last method again is what is termed as the labeling with a non-radioactive marker. So there we were using radioactive markers which is going to give rise to a particular detection. We can detect this radioactivity. However, there are some of the non-radioactive markers as well. For example, the use of biotin-avidin system, in which to the single strand or the double strand we have added what is termed as the biotin. These are all chemical molecules. Now these can be detected by the corresponding avidin molecules. So when we add these biotin molecules and when we add the avidin molecules, then they create a kind of a complex and there has to be some kind of a chemical thing. So there is a color change that can occur, or it can also give rise to a particular signal which can be there. So this is a non-radioactive marker labeling. And the last method is for example using horseradish peroxidase. Now this horseradish peroxidase again tags the DNA and it will give rise to a light intensity. So because when we add luminol, again a chemical compound, then when we use it is going to give rise to the light. And this light wavelength can then be detected in particular instruments that it is given rise. So wherever this label is going to bind, it is going to give rise to a intensity, a chemiluminescence which is called as. So because of the chemical reaction, some kind of luminosity is produced. So these are the methods for labeling which is basically used to label the probes. We will continue with the blotting techniques in the next session.